All right, so we are live on YouTube as well. Yep.
I hope you're all putting on your face mask because we are in a meeting.
Ha ha ha. I know, right? That's a good one.
Can't see your face mask.
Yeah. What's in there?
I think Henry has also joined.
Yes. Hello, Mr. Henry. You are welcome.
So I'll be back in about a few minutes, about 5 minutes. Is that okay?
Yeah, that's fine. That's fine.
Okay.
All right. So we are live on YouTube as well. And we are also live on Zoom. Hello, Henry. Can you hear me?
Hi, James.
Hello, Fifi.
Yeah, I have to drop. I'll join in later. So I have to catch up with something, so I'll join back later.
Right.
Bye.
Hi, Imelda. Thanks for joining us.
Thank you.
Please, which country are you joining us from?
From Mozambique, Maputo section.
Wow. Thank you so much for joining us, Imelda.
Thank you.
We'll be starting in a jiffy.
Okay.
Right.
Okay, James, I'm back.
All right. So once Prof is in, I'll just give the introduction, then we can start.
Okay.
So a brief background of SP and what we do. By the way, we are live on Zoom as well as we are live on YouTube, the SP Ghana section channel on YouTube. So if you can't join us on Zoom, you might as well join us on YouTube. So just a brief background of what we do and who we are. So SP is the largest individual member-based organization serving managers, engineers, scientists, and other professionals in the upstream segments of the oil and gas industry. SPE, meaning Society of Petroleum Engineers, promotes the exchange of technical knowledge within mainly the upstream oil and gas sector. It doesn't mean we don't have members in the midstream and then the downstream. In Ghana here, our members, some of our members are from Ghana Gas, which is a midstream company. So some of our members are also from the downstream sector of the industry. SP was established in Ghana in the year 2010 September, and it was through the magnanimity of Mr. Henry and some few industry giants back then. We have been at the forefront of exchanging technical knowledge, promoting the energy industry, and giving back to society. We have two types of membership. We have professional and students. So if you are a worker here and you want to join, just get in touch after the presentation and I'll get you enrolled. Somewhere March 2020, the WHO declared COVID-19 a pandemic, a global pandemic of immense proportions, affecting every aspect of the oil and gas industry and other industries. Oil prices dropped drastically due to the pandemic, and it has led to so many devastating effects on individual lives as well as families. Key to this is the COVID-19 pandemic. All right. So once you are able to know what the virus is, the good, the bad, and then the ugly, you should be able to also you might as well be able to know how to manage yourself and then stay clear of the virus. So therefore, the SP Ghana session through Mr. Henry Mam, Celestina, and then Riverson decided to put together this virtual technical meeting for us to talk about the COVID-19, the good, the bad, and the ugly. And we are also fortunate to be joined by Professor Reverend Professor Kwamina W.C. Segu. He is from the medical microbiology department of the University of Ghana Medical School. So without much ado, I'll hand over the baton to Professor Segu to take us through his presentation. Thank you very much.
Thank you very much. Can you hear me?
Yes, we can hear you.
Because we are doing a presentation, I want to make sure that you all put on your mask on your nose and your mouth so that we don't infect each other.
That's a good one.
I will skip some of the basic things, but discuss the issues at stake that are some being controversial so that we can keep abreast with it, and also to emphasize some of the issues that we need to take care of, especially as workers. But I first of all want to thank the SP, especially through Mr. Henry Mam, for giving me this opportunity to share some thoughts with you. So I'll follow this outline. Can everyone hear me?
Yes, please.
Okay, I'll follow this outline. I will first and foremost look at some characteristics of the virus, some basic things, transmission, and then just an overview of the clinical manifestations. And then we'll look at the global and national picture, where we'll look at numbers. And when we see the numbers, we will be able to understand what is happening. And then we'll look briefly at testing, but I want to put a bit more emphasis on the pool testing, since it caused some controversy some time back. And
I just log on. My friend sent that to me.
Pardon me?
Sorry, Prof. Just go ahead. Kindly, the audience, please mute your audio. Thank you very much.
So, and then we'll look about a few issues about drugs and some basic things that can help with prevention and treatment, like hydrogen peroxide prophylaxis. And then we'll end with the climax, the good, the bad, and the ugly. I don't think everything is bad. I think there are good sides to it too. You may find it strange, but we'll discuss some of these things as we go along. And I'll give you a few thoughts on some research that we are contemplating. And then we'll end with that. I put this picture there just to show something. Because it's a virus with an envelope, it backs out of our cells. So look at your cell, I'm sure a lot of us are guys. You remember we used to have this football, the Case 5. And then you had black patches and white patches. Now everything has changed. FIFA has colored the ball, so you don't see that. But just imagine that and shrink that in your eye or in your mind, and then see that as a virus, which will attach to something and then enter. So you can see the virus coming out of the cell. So the green surface is the cell and the virus coming out. So it quickly tells you the size of a virus. It is so small and so tiny that you need special aids to be able to identify this, to magnify it several times. You cannot use an ordinary microscope to magnify this. You need to use an electron microscope. So you have to magnify it millions of times, thousands, and before you can actually see the structure. So what is this virus? I mean, he's given James has given us an introduction. I know we've all remembered the Wuhan virus. Trump calls it the virus from China virus. But whatever it is, the basics we all remember and has not changed much. It's still the same story. But basically, the virus attacks the cells of the respiratory system and then gives you running nose, gives you a cough, sore throat, it gives you high temperature, and it is spread from humans to human. But initially, it's believed to have come from an animal reservoir. You call these things zoonosis. So they are zoonotic infections. But once it enters the human population and it establishes itself, then it can continue to spread within the human population. Now, if it enters through your nose and your mouth and infects the cells, there are some receptors called the ACE receptors. And when they attach to them, then it gives them the opportunity to enter your body. Viruses relate to the nature of viruses. You can relate to a lock and a key. If you have any good lock, you cannot use any key to open it. And so you need specific keys to be able to insert and then open the lock. If you make a copy of the key, you may not have that smooth opening as you would expect, like you would have done with the original key. So if you think of viruses that way, it gives you an idea of how they behave. Because they have proteins on their surface that they use in binding to the cell surface, the cell they want to infect, and that helps them then to enter the cell. If they don't have those specific proteins, they are very unlikely to infect that kind of cell. So with the ACE2s, it's found not only in the respiratory tract, but in other organs and even in our circulatory system as well. So it's widespread in the body, and I'll come back to that again. And so most victims may die from complications, including pneumonia. Now we are hearing stories of people having thick blood, people not even being able to breathe after they've tested negative, and so many things that follow after you've had an infection. But the pneumonia basically blocks the lungs and prevents you from breathing properly. So for example, a couple of months back, of course, when you have pneumonia, what happens is that when you have any respiratory distress, it means that your body is not actually, oxygen is not actually getting to your blood properly. So if you check your oxygen saturation, you can actually find out whether you are okay or not. So a couple of months back, I started feeling feverish and I thought, okay, this is it. So I tried to test. It was difficult to test because I was calling people and they were not responding. So I did my own home quarantine. So I bought a pulse oximeter and I was checking my oxygen saturation daily for a couple of days, just to make sure that even if it was that, my lungs will not be affected, because then I'll need some help from the hospital. So once oxygen doesn't get into your bloodstream, other effects can follow. Organs will die, your brain, etc., and other organs. I put this there just to make a point. I know that we are engineers, but you see, there's an interaction between the virus and the human body. And so when all these interactions occur, you have chemicals being produced, chemicals that control the body being produced. And these chemicals will ultimately control other aspects of the infection. So they call it a cytokine storm. When you're infected and you produce some sort of chemicals, the chemicals then help to destroy, as it were, the other organs of the body. So it's believed basically now that if you look at papers that are coming out, and there's a whole lot of information coming out, it is faster than anybody can cope with. But if you look at the information, it seems to suggest that when the people who die normally have this storm occurring. And that is why it damages their other organ systems and it leaves the respiratory tract and gets into the bloodstream. There may be many reasons why it does that. But two things definitely should be that. One, meaning it should be that it should have the receptors. It should have that kind of a key a lock and key relationship with the cells so that it can in fact infect more cells and then spread throughout the body. I got some beautiful slides from Worldwide Reach International SOS. So I use them as they are, and I've acknowledged them duly. I mean, I think they're beautiful slides. So now we say it's a novel virus because it's not been seen in the human population before. And it was first in Wuhan in China. And we're assuming that it started in from an animal reservoir. You know that in recent times they were saying that they had seen, even in China, they had seen some animals, some meat with the virus on. And so they started closing a few places down. We are still learning about the virus. We haven't finished yet. It's a long journey that we are going on. And now that vaccines are also being developed, I think the journey will even be longer than we think it will be, or we expect it to be. Now, most countries have the virus. And this whole thing started with travel, people getting into contact with people who had come from China and other places where they later on went to other countries. So this brought about all the controversies about lockdown and whether there should be a lockdown or whether we shouldn't lock down. But the whole thing about lockdown is that people carry the virus. So you want to prevent people from moving so that you can prevent the virus from spreading. We also know for sure that it spreads through droplets as you talk, cough, and sneeze. So the louder you talk, the more likely you are if you are infected to spread the virus. Or if you sing, you are likely more to spread the virus much more. And so the essence of using the face mask is to prevent that quantity of virus from coming out and hanging in the air or infecting people directly. Now, we believe it can enter the nose, eyes, and mouth. And touching objects like surfaces can also transmit it. So if somebody coughs and is not wearing a mask and you touch the surface and you touch your face, you can actually contaminate yourself. And that's where we should be careful. I'll talk about workplace infections. And that's a big issue about workplace infections. You know, familiarity breeds contempt. We know ourselves, so nobody wears a mask in the office, even if you are 2 or 3 in the office, because everybody is comfortable. And if anybody coughs onto his table and you're chatting and your hand goes on and you touch it and the virus is still surviving on the surface, then you're likely to be able to get it. So the workplace is a high risk area because one is that they are enclosed. The second thing is that people normally don't consider their colleagues or family members as people who would actually spread the virus to them. And so other people also are spreading the virus with minimal symptoms. And there's been a lot of debate on this, that sometimes people have symptoms before. So you may have a headache, you may have a short cold, but you recover. But that was the initial stages of being symptomatic, but your symptoms may vary. So people may decide then say that, well, even though you don't have symptoms, you've had symptoms before. So it has a lot to do with nomenclature, asymptomatic, symptomatic. But we've learned other viruses have shown that even when you don't have symptoms, you can transmit. And cytomegalovirus is one of them. And that's also something that spreads very quickly, especially amongst children who play in the creche, Sunday school environment. So it's not new for that to be said. People are there who are carrying symptoms but may not spread. There are several symptoms, and I've listed, you can see several of them on the presentation. Sore throat, cough, fever, shortness of breath. You are tired, diarrhea. That's why the toilet roll tissue paper got finished in the US, or they were struggling for and fighting over tissue paper. I mean, I believe that some of the symptoms they were seeing or had been told was that diarrhea would be a big issue. So within 14 days, I've seen some other publications where they've estimated that it could have to even be up to about 21 days. But generally, we've accepted that within 14 days after exposure, you can have the infection, or you can show signs that you have been infected, or you can can have an established infection. That is if you're asymptomatic. So people don't have symptoms or may have mild illnesses, but others may also have very severe conditions, which may actually even lead to death. I'm sure for those of us in Ghana, we've heard the ministers, a few people, politicians, talking about their experiences. So COVID is really real, and I hope people will listen. So now, with all these symptoms, there are less common symptoms. You have people who tell you that they lost their taste in their mouth, and somebody said, I just had a sore throat, or somebody having aches and pains, a rash. Those are kind of less common symptoms, but they exist. But the severe or the serious symptoms, the difficulty in breathing because your lungs are clogged, and then chest pain or pressure, and then loss of speech or movement. In fact, it's being said that now people are even getting stroke, a lot of stroke and things. I mean, like I said, the data is still coming out, and we'll find out with time what happens. Now, globally, as at the end of July, we have 17 million cases, 17,106 and 6,007 cases, and we have about 700,000 deaths. I deliberately checked at the beginning of the month, that is July, from the WHO updates, daily updates. And at the beginning of July, we had 10 million thereabouts, which means that within a month, we've added an additional 7 million cases have been added. Now, so that gives you an idea. If we can't control this, and this continues, within the next month, you can imagine the numbers of people who will be infected. So it's really a serious thing, and we need to find preventive ways. I think that should be the primary emphasis, preventive ways of controlling the spread of the virus so that the numbers can reduce. I put a few slides here from the Ghana specific data. If you go to the Ghana Health Service site, you see all this information. And you can see that if you look at the blue line, it's the cumulative cases we've had. But I agree with them that when you are looking at this data, you should also think of the fact that some people are recovering. And for people who are recovered or have spent the minimum time in quarantine or with the virus, they are less likely to transmit the infection. So the infection should be transmitted more with people who are active, have active infections. And I think over the month of July, we have not gone below 3,000 active infections. So if you look at the yellow line, it talks about the active cases. Active cases basically are people who have the capability of infecting others. And if you look around June 16th, you realize that there was a sharp dip, and then it seems to have gone to a lower level, and now it goes through, it's hovering around 3,000 to 5,000 active cases any particular day. And that was because of the change of the criteria for release or for recovery. First, we were doing 2 tests. You were tested till you were negative, and so recovery was slow. But in this case, they say once you stayed there for a minimum number, I think from 11 to 14 days, you are considered, depending on the nature of your symptoms you came with, you are eligible to be considered as recovered. So if you see this curve, I hope my colors are right. Is this orange? Anyway, the orange spots reflect the moving average, and then the new cases are the blue. You will find out that we peaked at a point with new cases. There are several reasons could have accounted for this, but I think one of the reasons also could be the fact that at a point we're having problems with testing. Things were a bit slow. So if you don't test early, what you do is that the people will stay in the system and spread it before they get their results. So that if the testing is not rapid and the results don't come in time, essentially what you are doing is that you are allowing people who are positive to continue to spread. But I would like to say this, I mean that I call this a global confusion. Every country is struggling in one way or another with their own problems. Some countries are doing better, yes, but many countries are still struggling. And I think as a country, we have done, I'm not saying it's perfect, there are issues, but we have done fairly well. I think we have invested a lot into testing and other things. We would have done better with more testing sites. But in the interim, um, I think that based on the capacities we have, um, this has done well. I have colleagues at Noguchi who are testing, including my own colleague who is here with me, but now is at Noguchi because of this. They, they run shifts and they are virtually working 24 hours to make sure that these results come out. Now, there's a common saying that it's only those with underlying problems that have issues. So people just say that, oh, me, I don't have any underlying problems, so I can do anything I want. I just picked the last two, I think last two updates for the death to show us that it is not only those who have underlying problems who are dying. Now, if you see 176, 177, 178, they are all males from Ashanti region. Now, nobody knows whether they have an underlying problem because it's not clear, it's unknown. Now, 60, 70, 60, it doesn't mean they don't have. Maybe with the circumstances, they could not know. Probably they died even before they got the history of their um, their medical history. But the point is this: I mean, it shows that we should all be careful because even people as young as 49 are dying, even though some have comorbidities. Now there's 181, a male, it's unknown whether the person had comorbidities. And the person is just 33 years old. I'm sure like most of us who are listening, or slightly older than him, maybe. And the same thing we have here. I mean, I've been informed that normally if you have three comorbidities, your chances of survival are low. Again, we can't also make conclusions because you will find out that there are a lot of people dying here in Ashanti region who are younger, and but you can't make any conclusion from this. I mean, these will have to the Ghana Health Service to carefully look at what it is. It could mean anything, but the point I'm trying to raise is that there may be several reasons accounting for these. But the message is clear that we all need to be careful. So underlying conditions have not been established in all deaths. And so we need to stick to preventive protocols, ensure that our work, church, mosque, home, where we are is a safe place to be so that we prevent getting infected. Now, I spend a little time on workplace infections because it's a big issue. Now, identifiable risk in the workplace includes surface transmission, aerosol transmission. And I'm extremely sure about what I'm saying, that the workplace is a relaxed environment. And I've seen it even in my own health environment, where people are not wearing masks and people are just comfortable chatting with themselves because they assume that, hey, these are familiar people. But you see, you don't know where your familiar people, familiar friends and colleagues have been to. You don't know the people they've come into contact with. You don't know where their family members have been and come home. So we all need to treat a workplace as a high risk environment, which has either surface, where we can actually get surface infections from surface contact surfaces, and then from aerosol transmission. And so some basic things that I'm not going to go through all the basic things that the Ministry of Health has highlighted. Those are standard things like washing your hands, using alcoholic sanitizer, wearing masks. All these are the basic sorry, all these are the basic things. But it will be good to keep alcoholic sanitizers at the entrances so that when people open the doors, because they are likely to wash their hands before they enter, so that when they open the doors, they can also use it before they enter. it's one step further, or you are likely to reduce some amount of hand contamination. And then we need to disinfect surfaces frequently, especially in places where you have a lot of people coming and a lot of people working, where there's a lot of contact on the surfaces. And as much as possible, even though I have not seen any major transmission patterns with surfaces. You know, respiratory viruses are respiratory viruses. They can actually get, they call them fomites. When objects are contaminated and you touch them, you can actually infect yourself. So as much as possible, we should keep away from sharing objects and sharing different things in the office. Let's stick to what we have. Let's sit well social distancing. Because of the aerosol transmission, I personally, I think that the recommendation for any indoor activity should be a minimum a KN95 mask, because the KN95 mask covers your entire mouth and it has filters on the side, not the nozzle, but you see lines on it, and that is because of the aerosol transmission. In my personal opinion, I think that is, if we are having meetings indoors, that should be something that we should all use so that we don't, because the mask is not foolproof. So if we wear masks that fit well and use filters or filters on the side, we are likely to protect ourselves and others if any of us is infected. And then the ventilation issue is paramount. I love meetings outside. I went to a radio station, I was invited to talk. And I told them, I'm not sitting inside. So we had it outside in the compound of the station. I thought it was perfect. I mean, we should encourage ourselves to use very well ventilated places with the fans on, with the windows open, so that if there are any viruses, COVID viruses hanging in the air, they are likely, the flow of air is likely to help to reduce the concentration which will be standing in the room at any given time. Some of our buildings have problems. There's no way you can have, and then let me say this. So we should minimize or possibly even stop using air conditioners if it is possible, but rather allow fresh air and ventilation to um, to be more effective. Um, there are some people who have gotten these humidifiers. If they are evaluated and they are found to be good, where you have some sort of a mist in the air, if you are in an office with somebody where you can do nothing about because you have to share that small, relatively small office, then what I will recommend, sorry, what I will recommend is that it will occasionally be good to use these humidifiers so that they, from time to time, they can actually also saturate the air. And in case there are any particles hanging, they could be dealt with. Now, so we, there are two major ways in which you can detect the virus. You can detect the virus itself or antigens of the virus, or we can look at how the virus responds when we get infected. And that is what we normally refer to as serological testing. I'm sure you've heard on the radio people making a lot of noise that we should do serological testing to see how far it has spread so that we can know what to do. Yes, it makes scientific sense. Unfortunately, they said most of the ones that they've evaluated at the Food and Drugs Board did not meet the minimum criteria for being used. So before we talk about this, there's also that language out there where people are talking about the type of laboratory. I'm sure you've heard they say P3, P4, high containment, etc. The level of the laboratory tells you the level of risky jobs you can do. So if you have a very low level laboratory, you don't want to go and play around with viruses which can easily fly in the air and then get you infected. You want to do it in a safe place. So most of the testing is being done in what we call level 2 containment laboratories. The idea is to contain things so that the people working don't themselves get infected, and then others also don't get infected. Level 3 is maximum containment, because you can actually grow viruses in a level 3 containment, especially for research, etc. So I know for now that there is a level 3 now in the new hospital that has been built. Noguchi has a level 3, and I know that the veterinary services also has a level 3. If I am right, I think Noguchi now has a level 4. So they are the only people in the country who have a level 4 at this time. But the complexity and the risk involved in the work increases with the level of work that you do. So for the normal testing that is being done now for testing for the virus genetic material, most of them are being done at level 2 laboratories, enough to contain whatever they would bring out there. So the other name that has been bundled around is the real-time PCR. It is like the machine is like a small printer in your house, but you connect it to a computer and a screen, of course. The reason why it is also referred to as a real time is that okay, let me take this first. So you see the viral genetic material is so small in the samples that are taken, whether it's a throat swab, whether it's a nasal lavage sample, whether it's saliva, but sometimes the virus amounts may be small. So when you talk about PCR, polymerase chain reaction, what it means is that if there's a little amount of genetic material of the virus, you can create an artificial environment where you can actually let it multiply so that it becomes several copies. So assuming that you had only 2, for somebody who was recovering and so did not have a lot of the virus in his throat swab sample, but because this thing is an artificial way of growing it and making more copies of the genome, the viral part of the viral genome, you're able to do it, and then it will be detected on a screen. That's for real-time PCR. So you will see that if you look at the copies, most of you are engineers, so you will understand 10 to the power 4, 10 to the power 5, 6. It's actually with increasing concentration. So you realize that as the virus is replicating, it gets to a point where a signal is given that the viral genome is present. And that is when it becomes like a sigmoid curve. It raises up and becomes a sigmoid curve. So if you have a lot of the virus in there in the sample, that curve will go up first. And if you have very little virus, that curve will come up later. So for example, the red shows that the person has a very high concentration of the virus. The yellow on the yellow, which is just horizontal and does not rise, is actually a negative sample. So the amplification could not pick anything. Sorry, but the yellow in the middle gives you an idea that that sample has some amount, but it took a lot more cycles of amplification for it to be able to come. So if you have a lot, it doesn't take too long for it to show. If you have little, it will take a longer time for it to show. And that's when they talk about the CT value, the time that it shows on the screen of the computer that you are positive or you are negative. Another thing that hits the airwaves, which even became political, was about the pool testing. Pool testing is not a new phenomenon. Now, if you have an infection which is going around and you have few people who have it, you don't want to waste money testing every single person, because the likelihood is that majority of them will not test positive. So because there are few in the environment, what you do is that when you get the samples, you put them together in pools of whatever numbers are appropriate. And this you can only do after you have evaluated the sensitivity of your real-time PCR system. So based on that, you'll be able to know whether pools of 5 will be okay, whether pools of 10 will be okay, whether pools of 15 will be okay. So assuming that you test people who are, okay, so 6 here, you see 6, 7, 8, 9, 10, all of them, none of them is positive. And so when you do the final test, everything becomes negative. It means that the whole batch is negative. But if you do this, look at the one at the top, one of them is positive. So once the pool becomes positive, then you have to test every individual sample in the pool just to be sure that, just to be able to identify the particular pool that, or the particular pools that made the whole sample, the whole mixture positive. So if you look at this, it helps to explain it better. It's cost effective in that if you test them in batches, the chances are that many of the batches you test will still be negative. So you don't have to test any individual sample, but a few of them may test positive. And for those ones, you will test them individually. And that's why Noguchi was testing so much samples. And unfortunately, people who were not knowledgeable were on the radio discussing it and saying that, no, it's not possible. If they count the numbers, it's not possible that they could have done this number of tests based on the output of Noguchi. But the fact is, if you use pool testing, you're maximizing, you can do thousands within a short period of time. But if the numbers keep rising, and you weigh the testing and you think that now more people are testing positive, it may not be cost effective to do the pool testing. It may be okay just to go ahead and do the testing in itself just like that. So you see this one too, one pool test positive. So you have the first round, you have 3 tests, one becomes positive. And then that one that positive is tested. So you see that only one of the pool becomes positive. And then the next one, you have one pool, and then the one is identified. For some of them, they may be tested individually. But what you're basically doing is you're reducing the number of testing that you are doing. This is also a similar thing. I'll skip this slide. So now let's look at what OECD says. I mean, they said an effective strategy that tests suspected cases, tracks people infected, and traces their contacts will help to reduce the spread of coronavirus. Now, if you test that somebody is positive, you want to remove the person, and that's why they were using the Pentecost Conventional Center because they wanted to move them out of this. They wanted to prevent further spread, so we just keep them isolated and then let them not spread the virus. So it's TTT. So you remember the curve I showed you for Ghana where there was a leap. If you are testing and your results are not coming early, what you're doing is that you allow the person to stay in the system for a while. That person is going to spread it to others before they find out the result has come. So if you test early and your results come early, and you get that person to isolate early, you are going to reduce the number of people spreading the virus within the community. Then they also say that to reduce the risk of a new second wave, outbreaks of the coronavirus, 70 to 90% of all people infected, persons come into contact with, need to be traced, tested, and isolated. So majority of the people who are infected, if they are isolated, will reduce the amount of spread in the community. And so it will help us to be able to reduce numbers. And this means that you have to have a huge increase in testing to be able to do this. And the challenges and cost of doing this will pale out to another lockdown. Now, there are controversies about lockdown. I mean, I'm not sure if lockdown is the most effective way for us. Even though it's effective, the dynamics of our country make it more difficult, because even if you look at the settlements that we have, there are real challenges, and there are real challenges. But even in countries which also did the lockdown, now they are seeing that they are having occasional spikes in specific places, and some of them are actually even locking down those places again. So it's not as straightforward as a simple thing that, yes, once you lock down, the problem is solved. It should come with aggressive testing, contact tracing, and the removal of people from others so that the infection does not spread. Now, one controversy that has hit the limelight is the use of hydroxychloroquine. I'll just leave it at that. There are arguments for and against. But it seems as if people are saying that if you start using it early, you are not likely. It serves as a prophylaxis. Others are saying that it is not helpful. In fact, I've actually heard somebody saying that it's actually killed people. Um, the other one is also dexamethasone, which is actually an old drug. These two drugs are all old drugs, and I'll make a statement at the end when I finish. These two drugs are all old drugs. Now, this group that I've cited here, Christian A and al., have actually come out. They've written a paper, and that's what I've cited there. Now, chloroquine is toxic. Apparently, the very dose that we take, I have malaria. It's a high dose. Chloroquine is toxic, but hydroxychloroquine is not as toxic as chloroquine is. And so you can afford to use low doses of chloroquine for short periods. But on the other hand, you can use hydroxychloroquine for a bit longer comparatively because of the nature of its toxicity. And they all have some sort of antimicrobial activity. So this seems to be the rationale for the use of hydroxychloroquine in the treatment of COVID patients. So this summarizes the various actions of the action of chloroquine. So chloroquine is an old drug. So, if a virus is replicating, what happens is that it first attaches to, so you see viral here. So let's say this is the virus. The virus then attaches. So these are the ACE receptors. Chloroquine has the capability of reducing the amount of viruses that enter. They also have a possibility of affecting the different processes that can help the virus to grow successfully in the cell. So that's why people are talking about hydroxychloroquine being used for treatment. Ghana has been using it. I am aware that they've done a clinical trial, and I got a hint that they may change our protocols as to where it will go to. We wait to see where it will get to. And then dexamethasone, these are old drugs, steroids. But the reason why people were hesitant for its use is that it can actually inhibit effective immune modulation. And the other thing also is that it can the other thing is that it's so immune modulation, meaning that it can affect antibody production, it may affect your T cell responses. As human beings, the things that could help to protect us when we come into contact with a foreign organism or microbe are basically in two arms or three arms, that which naturally God has given us. And then the ones which come in response to our body getting into contact with the virus. For that matter, we're talking about the virus. So it will produce some chemicals that will prevent the virus from growing, or it will produce the chemicals that can also, there are two chemicals, the antibodies that can neutralize it, and other chemicals that can control other factors called cytokines that can control other factors that could help you either recover or get worse. So dexamethasone is supposed to help us to the critically ill people. It's not for anybody, so it cannot be taken as what do you call it, prophylaxis. Like, so because I don't want to get sick, I'm taking it for a long time because that can actually prevent you from producing the necessary antibodies when you come into contact with the virus or the necessary chemicals to fight the virus. So yes, it can be used, and some reports are showing that it is helping to save lives. I also want to talk briefly about vaccines. A lot of people are banking their hopes on vaccines. I'm a virologist and I know vaccines have helped. I mean, my children have been vaccinated. I have been vaccinated for various things. So vaccines work. But It'll be important for us to be able to know what is happening on the landscape now. As at the end of July, I went to check the WHO site, and there are 26 vaccines in different stages of clinical trials. Stage 1, stage 2, stage 3. Stage 3 is the highest, where they test large populations to see how effective it will be and whether it really does protect people if you have to be vaccinated. And then there are other 139 in preclinical evaluations. So these are basically being done in the still worked on in the lab to see if they are available. Sometimes we have viruses that are not harmful. So they can grow in the human being without harming you. What happens is that if you take that virus and you genetically engineer it and insert part of the COVID genome that can produce antibodies to protect you, they can actually use it as a vaccine, so that they will use that to vaccinate you, so that then your body can produce antibodies to it, or they will use that to introduce the virus into your body, so that your body can respond immunologically to it as a way of protecting you. Some of them are live attenuated. In other words, the viruses will actually grow, and they have been modified in such a way that even though they grow, they will not cause disease, or they have been genetically engineered to fit into a bigger picture of a life system that can be used to introduce the virus into your body. Others are inactivated. I mean, they are completely killed. So when they are introduced into your body, all it does is that your immune system recognizes it and produces immune responses to it. And live attenuated vaccines are normally much better than the killed, or more potent or more effective, because you need very little doses because they will grow in the body. But for the killed or inactivated, you have to prime the immune system sufficiently to make sure that it can produce the necessary immune responses to protect you. And then we have what you call virus-like particles that look like viruses, which are produced in various ways. And then you can also use they're also using the genetic material of virus itself, or parts of the genetic material. And some of them are using DNA that has been coded with good information from the virus to be able to produce the needed immune responses to protect individuals. There are also protein subunits. You are designed it such that the proteins that will be produced is only part of the virus that is relevant. For a lot of them, they are using the spike protein, I believe. The spike protein is what generally can be neutralized or can be stopped from preventing infection of the cell or entry into the cell. These are the top most candidates that I saw at the WHO website. And all of these are in the, I think, stage 3 clinical trial, the last stage of the clinical trial, where they are testing large populations to see how best the vaccines will work. My candid opinion, I'm not hopeful that this year we will see a vaccine, because you have to follow up these vaccines. You want to make sure that you check for side effects. You want to make sure that these vaccines are not rather going to make more harmful to the individual when they come into contact with the live virus, because it's happened before for other viruses, where the people became the morbidity because they had been vaccinated and come into contact with the wild type, the actual virus, seemed to be very severe as compared to even coming into contact with the virus wild type itself. So there's a lot of safety issues when you're using vaccine, and that's why the clinical trials are important. So we would see. I mean, we will just have to follow the narrative and see what will happen in the next couple of months. But in my own personal kind of opinion, I'm not sure if we can get something before the end of the year. To think of the fact that we also need to produce a lot of them, a lot of doses to take care of the teeming people who need them. Now, I'm teaching, or I've just finished teaching. I taught some students from the master's students from the University of Cape Coast, Masters in Infection and Immunity. And then I was teaching them bioinformatics. Basically, bioinformatics is using computers to try to analyze the genomes, for my purposes, to analyze the genomes of viruses to see what information we can get out of these out of this analysis. So I just told them that, look, since SARS-CoV-2, which is causing COVID, is the virus, the topical virus, let us use that. So I limited them to the spike protein. The spike protein is where, if you're producing a vaccine, you should be concerned, because if there are changes, it may affect the effectiveness, how effective the vaccine will be, or it may even affect the testing kits that we are doing, among other things. So the spike protein is extremely important when it comes to how the virus grows in the host and causes disease. And changes in this spike protein can affect anything. Can affect the way the virus is spread, can affect the way the virus is virulent. Virulent meaning that how aggressive it is causing disease. It can affect several things. So we tried it, and I just did a few with them and gave them the work to do, to do some more work. And the interesting thing we are finding is that we are finding many mutations. And they've kept searching literature. I asked them to search. They say they still don't find. At least they've seen one which has been reported. The truth about science is that when you have sequences, you can actually put them in a database, a public database, which can be used. The only thing is that people should make reference to it and cite it. But sometimes people put sequences there and they don't just analyze them. And I guess in the heat of the moment, people are putting things out there. But it's interesting. And so I've highlighted, if you see this black highlight here, the D is for the amino acid, aspartic acid, and the G is for glycine. So obviously, at that position, you realize that there are changes that are occurring. It doesn't mean that the changes will immediately translate into a problem. But with time, if these changes continue, they may gradually have implications for other things that we do. So, for example, even for the vaccines that are being produced, if you have some kinds of vaccines, they may not be suitable for some kinds of people. So you're looking at a big picture of everything. And so these are called, we normally refer to these changes as mutations. And if these mutations continue, I know that it's been stated somewhere that there's been about 32 mutations or something that have occurred. I think I saw somewhere. But we checked and we were finding whether it was published. We weren't seeing it. But anyway, that's research for you. But these mutations are obvious that the virus may change from time to time. And it may help for the virus even to weaken, so that as it goes through the population, it will gradually itself find its way out of the human population and occasionally pop up somewhere or otherwise. I'll end just now. For this, what do you call it? You remember I talked about dexamethasone hydroxychloroquine. Now, I'm going to talk about hydrogen peroxide. Now, hydrogen peroxide is known to kill coronaviruses. So even a concentration of 0.5% can actually inactivate the virus. I want to make a disclaimer here that what I'm saying is not a national policy. So that if you are quoting me, you can quote me, right? But you see, the virus attaches to receptors. Before the virus can descend into your lungs, it has to go through your nasal cavity or your mouth, or for that matter, your eyes, to be able to come down and enter your lungs. So since hydrogen peroxide is used as mouth gargle by dentists, and this gargle can actually give some, I mean, technically, hydrogen peroxide can inactivate the virus. the thoughts that we have, I was working with another group, that if you gargle hydrogen peroxide where you have been exposed, you probably can prevent yourself from the virus from establishing an infection that is entering your cells to be able to grow, because it will have to attach. So when you get exposed and the virus has attached, once you gargle continuously, not continuously, I mean you gargle maybe 1% hydrogen peroxide 3 times at the same time continuously, and then maybe twice a day or thrice a day, you may essentially be able to inactivate the virus and prevent it from growing in the cells. Once it doesn't grow in the places where it entered in your upper respiratory tract, it cannot descend into your lower respiratory tract to give you more problems. So it looks as if that the old ticks at least are showing promise. That the 2 drugs, we haven't done a clinical trial yet, but I'm using it occasionally. But we are looking for money to do this clinical trial, because we believe that if we can prove that hydrogen peroxide can actually do this, then we can have a national policy on the use of hydrogen peroxide to limit people getting to limit people getting new infections. Yeah. Just 1 second. Okay, thank you. Sorry. To limit people getting new infections. So we are desperately looking for money and support. And I've told Henry already. And because of the nature of the testing issues, we want to see if we can set up our own testing facility, so that then we can use that to monitor the hydrogen peroxide. We're trying all sorts of places and people. And this is my appeal to you. For those of you who work with people who can support in those small, you see, the thing is, we should not rely on only a few places to test. And I'll talk about something as we go on, about the good, the bad, and the ugly. Now, the good is that it's preparing us for the next pandemic. So now we are desperately looking for ways of upgrading, for example, in particular, my lab, our equipment are obsolete. We need to find new equipment. Once people start building this capacity of being adventurous and trying to go into vaccines and testing and developing new assays, like you heard those in Kumasi were trying. It didn't pass, but at least it's a step forward. People are now being adventurous and want to take the bold step and do it and try it. After all, you lose nothing if you try it and it doesn't work. You learn the number of ways you don't have to do it. So we basically are preparing ourselves for the next pandemic, especially as we have seen that if that a pandemic comes, you will be left alone to cater for yourself. Nobody is coming to sit here to come and control anything to help you. And health-wise, we are building capacity. We are learning a lot. I believe that the doctors are now being innovative. I'm not saying they have not been innovative, but people are now thinking out of the box to try to do things that we usually have not done. And I think this is good training, because the next time the next one comes, we would also apply the lessons that we have learned from this. We are understanding viruses now. Almost everybody in Ghana is a virologist, which is okay, because now we know when you talk about viruses, you know that people understand what you're talking about. And in terms of novelty, we are looking at education and meetings. Different things are happening. Now people know that they don't have to rush in traffic to get to work, because they can actually start working before they get to work, even if they have to get to work. It has other implications though, but these are good things that you can see. Of course, bad things, the economy, where people are even threatening not voting for people because things are hard. Jobs will be lost, and already jobs have been lost. It reduces productivity. Many countries have had their GDPs dip. Controversy. Hydroxychloroquine, a typical example, and even vaccines, there are still fights. As remdesivir, another drug, which is so much controversy. In the midst of the pandemic, we are still having controversy. But the worst of it, or the ugly face of it, is that now people are having some sort of difficulties even after they have recovered. Some are dead, others are having strokes. Some of them have reported having thickened blood. And if your blood is thick, it affects how you function. Others are having permanent disability in their lungs. So, yes, it is bad, it is ugly in some sense. Maybe there's a particular group of people, but generally the information I've shown you shows that there's also a minority of people who may not have any underlying conditions, may also have problems. So I'm ending with this slide. And I've talked about the hydrogen peroxide already. We want to look at the efficacy. We have concepts we want to develop with vaccines, and we need support to do that. I have done some work with peptides and serological testing when I was in Sweden, and there's ways in which you can actually use them to be able to detect to work against cross reactivity, because some of the reports coming out are that the serological tests are cross reacting, they are not specific. But I use that for HIV-1 and HIV-2, where we use that to actually try to separate the antibodies from the two viruses, so make the testing as specific as possible. So we can always extend that knowledge into this. And then also to set up a peptide research lab to be able to do some of these things. So I'll end appealing that if you know any people who can support us in any way, my concept is that there should be basic equipment in every lab to make it possible for people to try their concepts. There are some big equipments that you don't need, that you can keep them in the big places like Noguchi, Wabip, and things, because they are difficult to maintain. But I think every lab needs some basic equipment. Thank you all for listening. I put the dog here because I read an article where they said a dog had gotten coronavirus infection. It tested positive. So that's just for your information. Thank you.
All right. Thank you very much, Doc, for such an enlightening presentation. From your outline, I think you covered most of the areas, and we are very much grateful. We'll take some questions. Let's first take some questions from YouTube. One person is asking, there is a suggestion that 5G degrades the immune system and that this has helped spread COVID-19. Is this true that this radio waves transmit these viruses?
Okay, my answer is simple. There are countries without 5G and they have COVID cases and they are dying. So that's the simple answer I'll give. There are countries without 5G who are dying. So I don't think we should encourage this. It's part of the controversy that is up the sleeve. There's no evidence for now, as far as I'm concerned.
Okay. Second question, he's saying that, second question from YouTube, Bentaki is asking, is there a correlation between one's blood type and their ability to be infected by COVID-19?
I have heard that this issue about blood types is coming up. I must confess that I have not looked at it extensively, but people are saying that the blood type may be helpful. But it stands to reason that because a lot of the effects are in the vascular system, in the blood, issues to do with the blood, there probably is. But I have to really find out on that. I need to find out and check the details on that.
All right. Henry is also asking, is there any explanation for the potency? Sorry, is there any expiration for the potency of this virus?
So like I said, okay, let me use this to explain. You see, influenza virus.
Okay.
If you are doing the vaccine, the live attenuated vaccine, you need to pass it through a system. You have to grow the virus several times in a different environment for a long time. Then gradually the virus becomes weak. And so when it becomes weak, then it can be used as a vaccine. So then it becomes weak in the sense that you may have mild symptoms, you wouldn't have severe symptoms. So as it goes through the human population and as these mutations are occurring, it is very possible that with time, the mutations would make it less virulent. By me saying less virulent, it's less likely to cause disease, but it will still spread. And gradually, it may find its way out of the population. But as it is now, looking at the numbers and the increasing numbers, like I said, from July, beginning of July till now, there's about 7 million. I think that it has no intention of leaving anytime soon.
Wow. All right. Another person is asking, can you throw more light on Ghana's recovery strategies? Formally, you need to be tested 2 times before you are finally discharged, right? When the recovery strategy or when the protocol changed, we added that once you are tested negative, you can leave. And then if you are also without a virus, you don't have symptoms of the virus, and then within 14 days, you are considered recovered, even without a test. All
Okay.
right. I really don't get that side very well. Being
Okay.
that I had the symptoms, and within 14 days, I'm not showing the symptoms again, and without a test, I'm being declared negative.
Okay, so what happened is, I know that in Singapore, I think in Singapore, a study was done where they realized that after 2 weeks, thereabouts, of being infected, if you're not showing symptoms, even though you may test positive, there are actually fragments of the virus that are found there, but it's not a virus that can grow. If it is not a virus that can grow, it means it will not infect you. So let me give another example. I was told that they took swabs from a few COVID patients who had died in the mortuary and had been in the morgue for about 3 days. And then when they tested them, they were positive from the throat swab. So the point I'm making is that the fact that you test positive, if there is any part of the genetic material there that the test will pick, if you take the swab, it will test positive. But it does not necessarily mean that you can infect someone because the live virus is not there. So the study that they did in Singapore showed that after a certain time, when they grow the virus could not grow, even though they were testing positive for the real-time PCR. And so it was all part of this that they brought out these new algorithms saying that rather than wasting your testing till the person becomes negative, because sometimes some people remain positive for the whole month, for about a month. They test after 2 weeks and they keep testing them weekly. They do about 3 tests before they become negative. So basically what the research is suggesting is that all those 3 tests that were done after 2 weeks, you may have found a fragment, but you may not have found the live virus there. So the chances of such people transmitting the virus is very minimal.
All right.
Is
Thanks,
it clear?
Doc. Yes, I think
Okay.
it's clear. It's clear. One person is also asking from the chat box that the isolation process, what is the minimum isolation process? You recall from the start of the virus, Ghana was isolation for 14 days. Even when guys were actually going on offshore trips, you needed to isolate for 14 days and then you finally go. Yes, as of last month, I think the protocols changed to just 7 or 8 days. And then if you don't show any symptoms, you are given the test and then you are gone. All right. So what is the minimum isolation period?
Between 5 and 14 days. I think a minimum of 5 days and 14 days. When you come into contact with the virus, you can show symptoms. Between 5 and 14 days, you can show symptoms. So I guess that there may be some other considerations they may have made to be able to make that decision that when you come from a trip, you stay within that period of time. But for those I know who came back, most of them, if not all, stayed for 14 days before they came out.
Okay. Okay. So will you suggest a 14-day isolation or a 5, 7, 8-day isolation?
If 5 is the minimum, I think that I would prefer a 14-day. But I guess that they took it in combination with other factors. The fact is, we have the virus circulating in the community already. They may have made other considerations, not just the time of a minimum period and a maximum period. Or they may have observed that from the data, and I don't have that data, they may have observed from the data of probably those who came back that most of them who were positive actually tested positive just after 5 to 7 days. After that, none of them became positive again. I hope you're getting what I mean. Hello?
Yeah.
Can you hear me?
Yeah, hello, can you hear me? Yes, I can hear
so
you.
then that becomes a scientific way. But like I said, I don't have the data, but it's possible that's what I'm saying, that it's possible. No, it's not possible. I'm certain that they would have considered other factors before
Other
determining
factors.
the 7. So, because it's very likely that those who came, the thousands of people who came, it's very likely that none of them or most of them didn't test positive after 7 days. That's one possibility.
All right. I'd like to put you on the spot here.
Yes.
Dr. Stella Emmanuel, a physician in Texas, USA.
Yeah.
She has been in the news for all the right or the wrong reasons.
Yeah.
She claims there is a cure for the virus. She says she has treated 350 people in her practice, and she used hydroxychloroquine. She said here in Africa, especially in Nigeria, where she used to be a physician before moving to the US, what she was doing was she was treating patients with chloroquine. So chloroquine is not a new drug. So she is stating emphatically that there is a cure for the virus. Do you believe her? If no, if not, what reasons will you give?
Okay, so if you see the slides I showed you, okay, I've seen another study from another review of papers from another person who has done a review. But the person seems to suggest that it is good for those who who are in the early stages of infection. The problem with Stella is that if you are presenting such a controversial issue, an issue which stirs controversy, it is important that you present data in a scientific way that will help. Nobody knows the kinds of patients that come to your clinic, whether they are severe, what kinds of patients they were, what kind of symptoms they had. So truly, you may have because it's an outpatient clinic. I think hers is an outpatient clinic. She may be seeing patients who are not coming with very severe conditions. And so what shows that it is the hydroxychloroquine that is keeping them alive, but they wouldn't have stayed alive anyway. There's definitely some evidence scientific evidence that the chloroquine issue is important. I wouldn't take that away from her. But I think that there's a need to present the data. Observational data is extremely difficult to interpret because there will be many confounders which we don't know. She didn't talk about the symptoms of these patients. She didn't talk about many things. She didn't have another group that she refused to treat. She was saying it was unethical because if the chloroquine is helping, why don't you treat? But the fact is, in the absence of that, it may be difficult to be able to make an emphatic statement. I am reliably informed that even the one which was used here, the hydroxychloroquine, they didn't see too much benefit in the hydroxychloroquine. Okay. So we need to, I think that what needs to be done is that all the data on hydroxychloroquine probably should be brought together. They should look at the patients, they should look at the setting, the kind of patients, and then they can evaluate it and see whether they're at what stage. Because at the moment, what comes to mind is that maybe the time of use may be helpful. We don't know. I mean, it has to be done dispassionately. Approach is everything.
Okay. Bentaki is asking from YouTube, is there a target date for Ghana to remove isolation and quarantine for us are even closed? Okay, Bentaki is asking, is there a target date for Ghana to remove isolation and quarantine for outsiders?
I don't get it. To remove what?
Isolation and quarantine for outsiders. So guys,
you mean guys who want to come back
to our country where they are required to stay? Yes. So is there a target into society or?
Okay, I didn't hear you clearly, but your line was breaking. But if I'm right, you're asking whether there's a time when they are going to lift the ban for people not coming in, travel ban. I'm not sure what.
Can you hear me?
Yes, I can hear you now.
Okay, Bentaki is asking, is there a target date which Ghana is targeting so that we will take away the 14-day isolation and quarantine period for those coming into our country?
No, I'm not aware of that.
Okay. Okay. Stephen is asking, and chloroquine itself, and which one will be better suited for Africa?
Well, I mean, so like if you cast your mind back, the slides, the chloroquine is toxic. That's why it can even cause blindness. So that's why when we take anti-malarials, we take it for a short period and we stop. So hydroxychloroquine, a bit less toxic, probably that's being used for treatment. So yes, chloroquine may be, but you can't use it for a prolonged period. So you don't want to kill yourself whilst you're trying to solve one problem.
All right. William is also asking from the chat box, have there been cases of transmission from animal to humans in Ghana?
Not heard of. Okay. But even the case I mentioned, I don't think there was an established transmission from the dog to the humans. It's just that the dog tested positive. So I'm not aware yet. But it's a zoonotic. So I guess the whole idea is that if the dog is infected and you are keeping dogs as domestic pets, technically there's a possibility, especially how people treat dogs in their homes sometimes. Some of them stay indoors, they lick around, etc., and things like that. But I'm not currently heard of any dog transmitting it to humans.
Humans. All right. As a virologist, look into the future and then give us when we should expect to see the end of the virus in Ghana, looking at our cumulative cases, our recovery cases, and then our active cases?
That's a very difficult question to try to answer. But the thing is that
Hello, Prof.
if you really
Hello,
want...
Prof. Can you hear me?
Yes, I can hear you. Can you hear me too?
Hello, Prof.
Yes, I can hear you.
Hello.
I can hear you. Hello. Can you hear me?
Okay.
Hello.
Yes, I can hear you. Okay.
I said that is a difficult question.
Yes, please go ahead.
But my point is that...
Can you go ahead, Prof? I can hear you.
Okay. If we really want to get ahead of the virus...
Answer.
Yes, I am. If we really want to get ahead of the virus, then our testing and the results coming back should be faster and should be within a short time. So once you test within the shortest possible time, the results should come. If we manage to go ahead of the virus and then keep all the protocols that we are supposed to keep, we will be able to bring the cases down.
Isso é estranho para os outros.
Hello?
Sorry, Prof, go ahead. I've muted the person.
Okay. So
I've muted the person, so you can go ahead, Prof.
Okay, so giving a time is going to be difficult, but the influenza pandemic 100 years ago lasted 2 years, didn't it? About 2 years. So we may have cases declining, but we'll still have the virus around for a while.
Okay. In the absence of, let me see. In the absence of any other question, Prof, I'll ask for your final and then departing words. And then we can bring today's meeting to a close.
So basically, with you who work, I know that workplace infections increased at a point. So it has become an important source of infection or spread of the virus. So my simple advice to all of us, let us not assume that because we have colleagues that we work with them, everybody is safe. Let us treat ourselves like aliens, but still be friends and colleagues and take the necessary precaution. And the second thing is that let us also eat healthy and get involved in activities that would help boost our immunity, so that if we come into contact with the virus, we may be able to fight it with our immune system. Yes. And then maybe the last is to make an appeal that you should support us to be able to do the studies with hydrogen peroxide that we want to do. So those are the things that I was going to say.
All right, Prof, one last question for you. William is asking the issue of mutation of the virus.
Yes.
Can you throw a little bit of light for us?
Okay, so part of the virus has proteins which help it to attach and to infect cells. It also has proteins that can be recognized by the human body and to prevent it from infecting cells. So if that part of the virus changes, it may affect some of these processes, including the use of vaccines and the ability for us to be able to test for antibodies to the virus. It is not every change that will result in this, but over time, as the virus grows and passes through the population, the community, the populations, it may still acquire these small changes over time that will change the property and the behavior of the virus. So the slide I showed you was one of such changes. But of course, it's yet to be established what that change can actually, the effects of that change. And also the fact that we should also have in mind that the fact that a change has occurred doesn't mean that something will change in the character of the virus. Is that okay? I was trying to look ahead here. So if you look at this, if you look at this, you see here.
Thank you very much, Prof. Yes, I think that's That should suffice. Okay. Okay.
Okay. Thank you very much for giving me this opportunity.
All right. Thank you very much, Prof, for your very enlightening presentation. I think we are now a little bit ahead of Ghanaian virologists, since within the presentations you said now Ghanaians are all virologists. So
Yes.
I think with this knowledge we've gleaned from you, I think we are a little bit ahead of most Ghanaians now. I also like to add my voice to Prof's appeal. He's saying that they need support to carry out research in the high.
Okay, I think it's done. Are you there?
Right. I think James may be having network problems at this point in time. Prof. Sego, we are grateful for your presentation, and he was trying to highlight the fact that you advocated for some kind of support to the research of the preventive drugs